at some point here. Okay, so continuing here with eukaryotic regulation of gene expression. We had stopped here last time that we had talked about Prader-Willi syndrome, right? And remember that I had mentioned 3 mechanisms that can lead to Prader-Willi syndrome, right? So the most obvious one is deletion of that piece of the father's chromosome, short arm of chromosome 15, but you could also have hypermethylation of a part of the chromosome that would lead to gene suppression, or you could have this thing called uniparental disomy. So you could have inherited 2 chromosomes from the mother. And here is the other example of imprinting that can lead to a syndrome, but in this case, you are missing the same part of that chromosome 15, but instead of missing from the father, you're missing here from the mother. So what happens here, also this type of genetic inherited problem is it's not very uncommon. So you have about 1 in every 20,000 births. Okay, so in this case, you end up with a syndrome that is totally different from Prader-Willi. So you're not talking about lack of muscle tone, and you're not talking about problems with feeding. But here, the problem that you have is that you have a child that is very happy all the time. So you have this frequent laughter. And you do have some uncontrolled muscle movements, but the main characteristic of this syndrome is a developmental delay. So you do have a level of mental impairment, and you have this continuous laughter. And you also have sometimes some seizures that are associated with this condition. Now, if you look at the phenotypes of this child with Angelman syndrome, first thing you notice, yes, of course, the child is always laughing for no reason. And usually the child has this big mouth. It's not just because the child is laughing, but usually the size of the mouth is bigger than normal. And so this type of this syndrome is also called happy puppet syndrome. So those are the 2 comparisons for imprinting, right, that you have. So you have your Prader-Willis and in comparison to Angelman syndrome. So remember that the causes of Angelman are the same causes as Prader-Willis, right? You can have deletion of the chromosome 15, but in that case for the mother, or you can have hypermethylation, or you could have uniparental disomy. In this case, the chromosomes would come both from the father instead of being both from the mother. So I mentioned also that the environment or your diet can influence your pattern of methylation, meaning proper methylation, right? So certain foods that you can include or keep in your diet are very good for keeping proper methylation of your genes. And you can imagine all the foods that hopefully your grandmother was telling you that you should eat. Those are the foods that you should be concentrating on. Mainly stay away from processed foods. Right, to stay away from sugar as much as possible, because sugar is not good for you, right? Except when it's in a bit of chocolate, that is good for you. Right? So good food that are good for you is broccoli or cruciferous vegetables. Okay, so those are very good at keeping proper levels of methylation. And eggs are very, very good for all levels of vitamins, and including vitamin B12. And some properties of red wine are also good for keeping a proper level of methylation, because wine has resveratrol. Okay, so here just FYI, what types of foods you should eat in order to keep your vitamin levels and overall DNA methylation in normal and good levels. Okay, so here's a question: why should you eat your vegetables? Okay. So mainly we have a lot of interest that sparked since in the 1990s, okay, related to methylation or proper methylation or improper methylation and development of cancer. And here you can see in numbers of publications since the 1990s that are relating improper DNA methylation with occurrence of cancers. And here I stopped in 2013, so you can imagine nowadays the number is even higher. So improper methylation will lead to development of certain cancers. So here is another study showing how your behavior can influence gene expression and could also influence whether you can recover or slow down the progression of certain cancers. So if you're interested in reading this type of study, what was done here was that they had patients that were suffering from prostate cancer, and they matched controls and experimental subjects, and they put the experimental subjects on a very particular type of diet, meaning I believe it was a Mediterranean diet, so very little processed foods, just natural sugars. It was not a ketogenic diet, but it was really avoiding a lot of processed sugars. And the other group was just kept on their own. And I believe the experimental group also had some meditation that was incorporated into their healthy lifestyle. And the results of these experiments were that the people who were put on a healthy lifestyle really had slowing down the progression of the prostate cancer. In some cases, they even had certain regression of their stage of cancer. So lifestyle can definitely change outcomes of certain diseases. So here is what I had just mentioned. Intervention group and then the results that happened. Okay. And this is a very interesting study that I had started to mention it before, but basically what this group wanted to test was whether transcendental meditation would help with overall concentration, recall and retention, and basically mental capacity, okay? So what they did here is that they put a certain group in a regimen of 20 minutes of meditation twice a day, okay? And the control group, what they did was that they placed the control group in 20 minutes of rest twice a day. That means this person would have to lay down and either take a nap or just not think about anything and just rest for 20 minutes twice a day. So difference here, these guys had to meditate, okay? Meditate, these guys had to rest. So here is your transcendental meditation group before and after the start of treatment. So this is their test scores before treatment, and here are test scores after, I think it was about a month of treatment. And then here is your control group that was just placed on rest twice a day, before and after the time period. And as you can see by the results of after, you don't really see changes between your control and your experimental groups. And what does that mean? Anyone awake? Data interpretation. With your skills to work. Sorry? Look at the graph. Does it show you a difference? Wow. It shows a difference before starting and after treatment. And then this is before starting and after treatment. If you compare this group with this group, like what's the difference? Not much, really. I would say that The control group even had a better result because they started at a lower score and they jumped to about the same score as the experimental group. That means that if you just take the time to rest, in this case twice a day, you can improve your mental capacity. So you don't have to meditate. Yay! You can just rest. You have to find the time to rest twice a day. But I would say if you rest 20 minutes once a day, maybe you go to half of that. If this thing is quantitative, it's better than not resting and just going crazy all day. Yeah. Yeah. Take a power nap, or if you cannot, fall asleep, just stay quiet and pretend that you're napping. That's considered resting. Yeah, isn't that difficult? It is not really difficult. I think meditation is difficult because when you have to think about not thinking about anything, that's a problem. But if you just rest and you want to relax, that's also very effective. Okay. All right. So that's your, clue for how to get your brain to work better without putting a lot of effort, right? And here are some other studies that you can actually look into this if you're interested, how your mind can control pain levels just with meditation. Yeah, I heard of people who do not want to take local anesthetics and they have to have root canals and they go to the dentist and just by meditation they can block tooth nerve pain. That's like extreme. I would never attempt that. But you can see that these people who are very much using meditation as a lifestyle, they start really early. I think this little kid must be like 3 years old and he is already meditating. You know when my kids would meditate at this age? I couldn't even get them to sleep at this age. Always on the go, right? So here is some examples of what meditation can do to your brain and perception of pain. All right, so here we go to the next stage in the book, so the next chapter, and we're going to start talking about membranes and fats. So things that you have to know. This chapter, when we're starting to talk about lipid biopolymer, so it is basically going to be a chemical that is made up of smaller molecules. And one thing to keep in mind when you compare other biopolymers, like for example, proteins or carbohydrates or DNA or RNA, you'll see that the lipid type of biopolymer it's not as complex, but it's extremely important for many areas in cell function and cell signaling. So what is the use here for lipids? Well, we can store lipids as fuel, and you can also, of course, you must have lipids to make their cell membranes. Every cell has this lipid bilayer. Right? You can use lipids to make hormones, basically steroid hormones. Okay, so your steroid hormones are going to be derived from cholesterol. You're also going to use lipids to make some types of soaps, like bile acids. That's also derived of cholesterol. Okay. You're going to make some signaling molecules, and you're going to use lipids also to derive some fat-soluble vitamins. Okay, so lipids are important for everything that you can think of in cell function. Yep. Yeah, that's one. Yeah, oil is a lipid. Yeah. So we're going to have to know certain properties of lipids, okay? So here, remember that certain vitamins are lipid soluble, so that means those vitamins are also lipids, and you remember what they are, right? A, D, E, K, or you remember that right? That's easier to remember than if you're going to remember the vitamins. So if you just put like vitamin naked, those are lipid soluble vitamins, right? So a lot of lipids, in order to be qualified as a lipid, you have to have a bulky section of this molecule that is just hydrocarbon. So to be a lipid, it doesn't mean that the entire thing has to be hydrocarbon. You can have some areas of the molecule that have polarity, okay? But then the proportion of this polarity to the rest of the molecule, this has to be smaller than the rest of the hydrocarbons that are present in the molecule. Okay. So here, if you look at this molecule of a fatty acid, you know that most of the molecule is hydrocarbon, and only a little part has this polar characteristic. Then compared to cholesterol, you only have a tiny, tiny part of polar characteristic, and the rest is hydrocarbon. So when you have a lipid that has some polar characteristics, but the rest is hydrocarbon, you call that an amphiphilic lipid. Okay? Okay, so we just mentioned that. Okay, so mainly they're made of hydrogen and carbon, and mainly you're going to have this non-polar molecule. So this non-polar part of the lipid is going to give you your hydrophobicity, and then the polar part is going to give you hydrophilicity. So here, key features of lipids. When you look at fatty acids, as you've seen, you're going to have a little part that is hydrophilic and a huge part that is hydrophobic. Okay, so looking at this type of lipid in relation to that type of lipid, you see that this guy here is a straight line. So when you have a lipid that is very straight with every carbon that is saturated, meaning every carbon is connected to hydrogen and you have only one bond between the carbons, so you call this lipid a saturated lipid. Okay, so saturated means that you have single carbon bond and it's saturated with hydrogen, okay? And when you look at saturated lipids, those tend to be solid at room temperature. So an example of saturated lipid is butter. Okay, so it's solid at room temperature. Now, when you look at other types of lipids that have a double bond with the carbon, those are your unsaturated lipids. Now, if you have one double bond, only one double bond, that is already unsaturated lipid. If you have more than one double bond, then it's also unsaturated lipid. So you need at least one double bond for this thing to be considered unsaturated. And unsaturated lipids, they tend to be oils at room temperature. Okay? So for example, here is olive oil. If you have it at room temperature, you're going to see that is a liquid. But if you put your olive oil in the fridge, then it's going to solidify. Okay? So we're going to have to know the relationship between number of double bonds and melting temperature. Okay, you got to keep that in mind. So one important thing here to consider is that all naturally produced lipids are cis conformation. It means this attachment of the carbon is going to be to the same side when you compare here with your double bond. Right? So most people do organic chemistry and you know what the cis looks like, right? In comparison to the trans, right? So that's cis. So let's go for a quick exercise of cis and trans. Cis, trans, cis, trans, trans. Ah, it always happens. Okay, so you gotta know what is cis and what is trans. Okay. Can you get naturally occurring trans fatty acids? This guy over here. No. How do you get trans fatty acids? Industrialization or high heating of certain oils will give you a lot of trans fatty acids. Can you consume trans fatty acids? Yeah. Can you get rid of trans fatty acids? No. Where do they go? Which fat? Like where? It will go, actually it will accumulate in your arteries. Plaque. That's why it causes plaques. And that's what leads to atherosclerosis, heart attacks, and all kinds of complications, right? Once you start accumulating trans fatty acids in your body, you can't really get rid of them. Right? Now, when you look at certain processed foods in the supermarket, make it a habit to see what are the composition of the foods that you're buying, right? Try to buy fresh fruits and veggies because, they're just better for you, even with the pesticides, just wash pesticides away. Now, if you look at the label of your foods, let's say potato chips, and you see 0 grams of trans fatty acids or trans fats, does it mean that it does not have trans fat in it? No. Trace, kind of not just trace, right? It means that the food industry is allowed to put 0 grams of trans fats per serving if the total amount of trans fat per serving is less than 1 gram. It's very sneaky, okay? So it is 0.9 grams, food industry can put 0 grams. Now, bag of potato chip will usually have 15 servings or more, right? Maybe a serving of a potato chip is like 1 potato chip. And who is going to stop at 1 potato chip? Doesn't make sense. If you buy a bag, in my case, I'm going to eat the whole bag, right? So in that case, you're going to eat about, if it has 15 servings, maybe 14 grams of trans fatty acids. Is it still 0 grams per serving? That is not good for your health, okay? So keep that in mind. Stay away from processed foods if you want to keep your cardiovascular system in good shape. And then, of course, also exercise and take rests. Yeah, it's getting a lot of lists here. Checklist is increasing. All right, things that you have to know about your fatty acids. If you have here stretched out, so this guy here is showing you a trans fatty acid. Is that naturally occurring? No, that's artificially produced. If you have here a cis conformation, that is naturally occurring, okay? Now, you look here, if you start to increase the number of carbons in this big tail over here, you start to give them names. Do you have to worry about those names? No. I always forget about those names. But you have to worry about whether they're saturated or unsaturated. So if you see a molecule that kind of looks like this, I don't care if you count the carbons or not, but it looks unsaturated to me, okay? So you look at the number of carbons and you're going to say, look at this, naturally occurring, it goes like 12, 14, 16, 18, 20, and you notice that every time you have 2 carbons, they get added. The reason for that is that the enzyme that adds carbons to fats, it adds 2 carbons at a time. Okay, so if you have naturally occurring molecule, you're always going to have, most of the time you're going to have multiples of 2 because that's how the enzymes will work. Okay. So now here, when you look at your unsaturated fatty acids, you're going to see at least 1 double bond. So you have to take in consideration when you look at property of fatty acids, whether it has double bonds and how long that molecule is, because those are going to interfere with very important properties of those fatty acids. Okay. So you mentioned here that fatty acids are amphiphilic, so they do have that little polar head, right? And they're always in this cis conformation. And here comes with the nomenclature of those unsaturated fatty acids. There are two ways to give them names. If you start at your carboxylic acid polar head, you're going to name this based on a delta type of numbering. So you have to count the carbons and figure out how many carbons are there until you get your double bond. So here, if you start a carboxylic acid, that's a delta form, and you start 1, 2, 3, 4, 5, 6, 7, 8, 9, and there's your double bond. So you say this fatty acid is delta 9 fatty acid. Now, what we are most used to is the omega type of nomenclature. In that case, you start at the tail that is hydrophobic. So now, let's see, if you count this oleic acid here, you're going to say 1, 2, 3, 4, 5, 6, 7, 8, 9. Oh, coincidentally, omega-9 fatty acid. Now let's look at some other ones. And you say here, if you go this guy, 1, 2, 3, 4, 5, 6. Oh, omega-6 fatty acid. But this guy has 2 double bonds, so it's like 6, 7, 8, 9. So this is 6 and 9 omega, right? Omega-6 and 9 fatty acid. Okay, so if you go from the other side, you'll see this is going to be delta 9 and 12 fatty acid. Then you would see this other example here that has 3 double bonds. It is still unsaturated. It's more unsaturated than those guys over here, right? But now here you're going to say omega nomenclature 1, 2, 3. Oh, this guy is omega-3. And we know omega-3s are anti-inflammatory, right? Omega-6 tend to be a bit pro-inflammatory. But you need a balance of those omega-3, 6, and 9 fatty acids in your system in order to get proper cell function. Okay? So some fatty acids that are needed for health and that we are not able to produce in our bodies, those are called essential fatty acids. Okay? And here is an example of 2 essential fatty acids. Okay, so let me emphasize that. So animals cannot make double bonds at delta 12 or delta 15. Okay, so here delta 12 or delta 15. Delta 15 here is in comparison to your omega-3s, right? So when you give names to those fatty acids, those are called linoleic and linolenic acids. So those guys that we cannot produce, those are your essential fatty acids. That means you have to eat them. Where do you get your omega-3s usually from? Fish. What kind of fish? Salmon or cold water fish. Okay? Now, this thing says animal cells cannot make those fatty acids. Are fish considered animals? Yes, where do they get the omega-3s from? From the food they eat.
They also are animals.
Hopefully those are going to eat some omega-3 fatty acid foods. It goes down, up the food chain. Okay? Basically, yes. Yeah, and some algae and, some bacteria or those plankton species. Okay? So you are going to get those from cold water fish because they ate it from somewhere else and they tend to accumulate it in their tissues. Be careful with farm-raised salmon. Yeah, try to get like free, wild caught type of fish. Those are better for you. All right. So here, so those are your fatty acids, right? And we're gonna find more about their temperature in a little bit, okay? Melting point, I meant. So here, talking a little bit about fats. So if you get 3 fatty acids and you do dehydration reaction using a backbone of glycerol molecule. Okay, so here's your glycerol. Okay, if you do dehydration reaction, you are going to end up with a triglycerol fatty acid. Okay. Triacyl fatty acid, triacylglycerol, that's the name of it. Okay, and this is your fat. Okay, this is a molecule of fat. So usually here you're going to have this molecule that is insoluble, is solid at room temperature, and of course all fats are insoluble in water. Okay, so this is solid at room temperature, and this is a type of molecule that you tend to put in fat granules inside of adipocytes, and those are your fat cells. Okay. So this is the type of fat that you use for storage. So here is where we start talking about melting temperature. All right. So first thing that you see here, if you increase the tail of that fatty acid, you are going to increase your melting point. So what does that mean? The longer the tail, okay, the longer the tail, the higher the melting point. Okay, so let's say if you get something like this, muriatic acid that has 14 carbons and they're all saturated. If you put it at room temperature, is this thing a solid or a liquid? Okay, I got solid over here. More votes for solid? Yeah, I got a few undecided votes kind of going like this. It's like, oh, it's a mistake. Okay, go for it. Solid. Who thinks it's solid? And how are you going to find out? What is room temperature? 45, 25 degrees Celsius. Equivalent to what? 75 Fahrenheit, right? Okay, so you look at this and you say, okay, melting temperature in Celsius is 58 degrees. Is that hot? That's quite hot. So is this thing solid at room temperature? You have to heat it up to 58 degrees Celsius to get it to be a liquid. So this thing is solid at room temperature. Okay. If this melting temperature was in Fahrenheit, this thing would have been a liquid. Okay, but this is in Celsius. Okay. Now let's see this guy here. Is this solid? Well, yeah, it's getting hotter and hotter for this thing to melt. So all these things are solid. Okay, solid at room temperature, but you got to put some heat in there for this thing to melt. Now, when you have 14 carbons, they're all saturated. You only need a little bit of heat. When you have more carbons, you have to add more heat. So the more carbons you have, the higher is your melting temperature. Okay, everybody got that? Okay, now let's see in comparison here to your levels of unsaturation. The more unsaturation you have, the lower your melting temperature. Okay, so this is an inverse relationship. The more double bond, the easier it is for this thing to be a liquid at room temperature. So let's see what happens here in this case of stearic acid, that is 18 carbons. Okay, this guy here is your saturated guy from here. Okay, same melting temperature here and there. Okay, everything saturated, melting temperature is 71 degrees Celsius. Okay. If you put one double bond in this 18 carbon chain, now your melting temperature is going to be 16 degrees Celsius. Is that liquid at room temperature? Okay, well, again, what is your room temperature? Room temperature is around 25 degrees Celsius. So if you're in doubt, what do? You are going to make a graph, okay? And you're going to put here, this is 25 degrees Celsius, okay? This is your room temperature, right? If something is under 25, it is liquid at room temperature. If something melts above 25, it is solid at room temperature. So let's see, this guy here, saturated is solid. Everything saturated is solid. This guy here, saturated is solid. If you put one double bond, now melting temperature is 16. So now it's liquid. Okay. If you put two double bonds, your melting temperature is, look at that, -5 melting temperature. At what temperature does the water freeze? 0 degrees Celsius. So if something has on the membrane a lot of this linoleic acid that has two double bonds, it can survive in freezing temperatures, right? Because the membrane is not going to solidify. So it's actually useful for this species to have in cold temperatures to have those double bonds. Okay. Now look at this. If you have 3 double bonds, negative 11 degrees Celsius. Okay, so the more double bonds you have, even keeping the same number of carbons, the more double bonds you have, the lower is going to be your melting temperature. Okay? And I always like to ask questions, putting in a stem that would give you a certain scenario for for some bacteria or for some fish that are living in certain weather conditions, whether double bonds are going to help this thing to stay alive or not. Okay, so you actually have to think about how you use this knowledge, okay, in these types of scenarios. So when you are studying for your next test, which is coming up like next class, right? Friday? Oh, I mean, yeah, Halloween. Yeah, how can I forget? Halloween is going to be your test, right? So Halloween, if you want to come dressed up in Halloween style, that would be cool. I think I'll come like dressed like a witch, because that would be just perfect. All right, witch giving test in Halloween. Yeah. Okay. So you have to think about what kind of questions I can ask about this thing, right? What is an animal that lives in a cold temperature in comparison to an animal that's living in warm temperatures, and how this knowledge of melting points of lipids is going to help the animal to survive. Okay? So use your AI to come up with questions. And here comes your first, oh, there we go. There's an example question. So we open this up. Okay, here are your iClickers. Is this thing connected? Is it working for you guys? Oh, now it's working. Okay. This thing says it's working. It's not working. The base is not working? Oh, the nightmare. Okay, a lot of people have already put in the answers. Video is not picking up. Let's see, tools. Okay, I'm going to try to stop this and start it again. You know, those glitches that we always get with this iClicker, such a pain in the neck. Okay, I'm trying it again. Let's see if it works now. All right, now I think it's working. Okay, so I'm going to cancel the last session. So everybody vote now to get your points. Okay, I'm going to cancel the last quiz thing. E. coli bacteria that is growing at 37 degrees Celsius, that's body temperature, right, is moved to 25 degrees Celsius. Which of the following changes must be made in the plasma membrane to help it cope with the decrease in temperature? Does anyone need more time? Everybody answered? Okay, I'm going to stop it. What is your answer? D. So here it says the number of unsaturated fatty acids must increase. That's your right answer. So here, properties of unsaturated fats. Of course, you must have at least one double bond. Okay. You're not going to have nonlinear chains, so everything. Okay, guys, let's be quiet here. So you're going to have everything stretched out. You're not going to have branches out of this molecule. Okay. You're not going to have interaction between the chains. So each chain likes to stay by itself. And fats will usually have low melting points, okay, or unsaturated fats, okay. Usually you're going to have them in the form of oil in room temperature. Okay. Properties of saturated fats. Only carbon-carbon single bonds. Those are going to be closely packed. You're going to have a strong interaction or attraction between the chains, okay, those van der Waals forces, okay. You're going to have high melting points, and they're going to be solid at room temperature. So we have seen this before. This just repeats. Where is it that you're going to find fat storages? Well, we know under the skin, so subcutaneous fat. Usually that is not as harmful as visceral fat. So visceral fat is going to be in your belly and around your organs, okay? And actually, some people who appear to be skinny, they could have a lot of visceral fat, and it's just that you don't see it, right? What do you call those people? Lucky? No. You know the term that those people like thin on the outside and fat on the inside? Kind of. We call it like tofu. Thin on the outside, fat on the inside. Yeah. Tofu people. Visceral fat is more dangerous than adipose, than subcutaneous fat. Okay, so use that to store energy, and let me see. Besides storage of energy, your fat tissue is also important for production of hormones, okay? So you're going to have hormones and other molecules that are used in signaling pathways. So the molecules that are produced by fat tissue, those are called adipokines, okay? And some of the hormones that are produced by fat tissue, that's basically your, not the ghrelin, leptin hormone. Okay, that gives you a feeling of being satisfied after you eat. So how is it that you're going to break down fat? Well, you made up this triglyceride by using dehydration synthesis, and you're going to break them up by using hydrolysis, of course. Okay, so you're going to have enzymes that are going to do hydrolysis, and they're going to separate. Okay, you're going to separate your fatty acid from your glycerol backbone, right? That's the part I want you to remember. This is a hydrolysis reactions, and you have enzymes for that. Okay? We're not going to go into details of names of enzymes and the order of where these chains start to break down. That's too much detail. But you have to remember this is hydrolysis reaction. So here, what kind of fatty acids that you can have and how is it that they are useful? So you can have some fatty acids that do not have a long carbon tail. So how short? You can have fatty acids that are very short. So they can have between 2 or 4 carbon in the backbone. So you call them short chain fatty acids. So between 2 and 4 carbons, those are put into a bucket of short chain fatty acids. Then you have some other fatty acids that they have a bit of a longer chain. So if they are between 6 and 10 carbons in this backbone, you put them in the bucket of medium chain fatty acids. And then if they have more than 12 carbons, you put them in the long chain fatty acids, right? So you can have short chain, medium chain, and long chain fatty acids. So you should know what's the number of carbons for each one of those buckets. Okay. All right. So here, you have certain short chain fatty acids that you do not produce, but you have your friendly bacteria that produce them. Okay? Where is the friendly bacteria? In your gut, in your intestines. Okay? So those friendly bacteria, they're going to produce these short chain fatty acids. In particular here, you have acetate, propionate, and butyrate. Okay? So it's like 2, 3, and 4 carbons. Okay? And what this, here's the formula or the chemical composition of those short chain fatty acids. Okay? And basically, the importance of those short chain fatty acids is that they're going to help your intestines to be healthy. They're going to make sure that this barrier between your intestine and your system is well maintained. So that means it maintains the intestinal barrier. It helps, these fatty acids helps with the production of mucus. Okay. So mucus is going to be right here at the lumen of your intestine. So the more mucus that you have, mucus is, let's put it a green. The more mucus that you have, the better your barrier protection. Okay. Now, besides that, these fatty acids are absorbed in your system, and they are going to cross this system and get in connection from your brain to your gut, from your gut to your brain. So this is your gut-brain axis, and it's going to get into your brain and help with neuronal functioning. It's going to help your neurons function better, and it's going to help your accessory cells like microglia and astrocytes. It's going to help your accessory cells to work better. Okay, so short chain fatty acids produced by your gut bacteria are very important for proper brain function. Okay? Besides helping you to slow down gut inflammation. Okay? So protecting you from gut inflammation, helping with brain function. Okay. Now look at this. This part is interesting. These two fatty acids, propionate and butyric acids, butyrate, they inhibit certain enzymes that are involved in the development of those neuropsychiatric disorders. Like what? Like depression. Okay, a lot of people suffering from depression these days. Okay, schizophrenia, even Alzheimer's disease. Okay? So basically, this is telling you keep your gut bacteria happy, and it will help your brain to be happy. Okay? Avoiding diseases in the future and maintaining a healthy brain, mental state. Another question. Right. Which of the following is true of unsaturated fatty acids? They are naturally produced in the cis configuration. They include stearic acid and palmitic acid. Unsaturated acids. Okay, does anyone need more time? No, everybody answered? Yeah, okay, I'm going to stop it. What is your answer? Hey, it's not getting, there we go. All right, so important things about those membranes, we have talked about those things already, right? So now, looking at the lipid bilayer, you're going to see what makes the lipid bilayer. And two lipids that you have to keep in mind is always the glycerol phospholipids and your sphingolipids. Sphingolipids are going to have a portion of carbohydrates that is going to be attached to them. I'm going to see what they look like. So you're looking at cell membranes. What you're going to find in the cell membranes? You are going to find these glycerol phospholipids that are going to make two layers. The fatty or the carbohydrate parts are going to be facing each other. It's going to be the non-polar interior part of a membrane. The outside is going to be of your polar water-loving part of the membrane. So this is going to be your phosphoside. Okay. So in the membrane, you're going to find proteins that are going to span through the membrane. So you call those transmembrane proteins. You're going to find some proteins that are attached either to one side or the other side of the membrane. So those are called peripheral proteins. Okay, so here transmembrane protein, those are your peripheral proteins. And you're going to have glycolipids that are sticking out of the membrane, mainly on the outside. Okay, and then you're going to have some receptors that are also made up of proteins that they span the membrane. The only that is outside, it's inside. So those transmembrane proteins, they're also called integral membrane proteins. Okay, so if it goes across, it's integral or transmembrane. If it is only on one side or the other side, those are your peripheral proteins. So here, what is it that can go through membranes? So this is the part that you have to start remembering things. Okay, but it's actually very simple. If you have small hydrophobic molecules, okay, or gases, those can just go through the membrane without any help from anything. Okay, so what are you talking about? Oxygen, carbon dioxide, nitrogen, small hydrophobic molecules like benzene. Those will just go across without any help. They diffuse across the membrane. What is going to control the level of diffusion is just the concentration of these things on this side in comparison to that side. Okay? So if you have a diffusion gradient, these things are able to go through without any help, just diffuse by themselves. Is that a good thing? Yeah. Think about oxygen coming to your tissues and then carbon dioxide being expelled from your lungs. So if you don't have simple diffusion, that could be a problem. Okay, so this is a good thing. Now, if you have some small uncharged polar molecules, you're going to have a very, very small gradient of diffusion, but mainly they're not going to diffuse. You're going to need something to make this thing go through the membrane. Even with water, you have channels, they're called aquaporins. They're going to allow water to go through the membrane. Water just by itself is not really going to go through the membrane. So let's see here. If you have large uncharged polar molecules, those are also going to be blocked from crossing the membrane. So the only things here that will go through the membrane, you have to keep in mind are small hydrophobic molecules and gases. Gases will go through without a problem. Okay? So if you think about bigger molecules, they will need help. They are not going to go through the membrane. If you have ions, those are small. Okay? Those are small, but they are charged. Ions are charged. They are not going to go through the membrane. And that's a good thing. Okay, so ions will not go through at all. unless you have a transporter that will go there and allow the ion to go through. Okay, so ions will not go through the membrane. Okay. So here, looking at the composition of the lipids in the membranes, mostly you're going to have phospholipids, okay? And you're also going to have some cholesterol that is going to be in this hydrophobic part of the inner part of the membrane. Okay, so you're going to have a lot of phospholipids, some cholesterol, and you're going to have other lipids that are going to show up and be integrated there. Okay, those are lipids that can have some kind of sugars on their surface. Okay. So the type of lipid that gets in there, like your phospholipid, it is going to be made of two of those hydrophobic tails and a big polar head. Okay, those lipids are amphipathic because they have a water-loving part and a fat-loving part. Okay, so amphipathic lipids, phospholipids. So here looking at how big the membrane is actually very small. This is about 5 nanometers. Okay, very, very tiny. Okay. And again, the composition of those lipids, you're going to find mainly phospholipids, and you're going to have some glycolipids and some cholesterol that is going to be inside of the membrane. Now, cholesterol, as you're going to see, is very important for cell signaling. It's also very important for viral infection and membrane vesicle fusion. Okay? And they tend to aggregate in a certain part of the membrane, and you call those aggregations of cholesterol, you call them lipid rafts. And they tend to be resistant to detergent dissociation. So let's see here. What is happening here with the membrane? Okay. So remember that if you have unsaturated fatty acids, you're going to have a lower melting point, right, in comparison to your saturated fatty acids. So here, if you look at a membrane that is mainly composed of saturated fatty acids, you see that those fatty acids are not very movable. They don't like to move around a lot because they're really stuck to each other in this type of conformation. Right? So the fluidity of saturated fatty acids is very low. Not a lot of fluidity going on here. Okay? If you compare that to unsaturated fatty acids, you see that there's more space between the molecules, and these molecules can move around a bit easier. Okay? So let me see. I think this was a little movie. Yeah, you see, those unsaturated fatty acids can move around and give more fluidity to the membrane. Okay? Stop. Okay. So let's see what is meant here by fluidity of the membrane. So when you look at these phospholipids, let's say here you have oil and you have water, so we have an interface over here. You know that polar heads are going to face the water, and the non-polar tails are going to be facing the oil. Right? So here, if you have this membrane that is a double layer of fatty acids, right, that is facing each other, you see that the polar head, it can move around a little bit, it can twist around, it can spin, right? And if you have unsaturated fatty acids, those can move more longitudinally, okay? But one thing they cannot do, they cannot flip from this side to the other side. So fatty acids that are on this side of the membrane, they stay there. Comparison to fatty acids that are on the other side of the membrane, they also stay there. They're not going to go flipping from one side to the other side. Okay? So, when you look at inside of cells, and you see that some fatty acids are actually flipping from one side to the other side. But they're not doing that by themselves. They're using proteins. So look at these cool proteins here. You have this transmembrane protein, meaning it spans the membrane on both sides, right? So that's an integral protein, right? And some of these proteins, their jobs are to flip. The job is here to flip this fatty acid from the outside of the cell to the inside of the cell. So you call that protein a flippase. You know, to give name of proteins, you put A's at the end. So this guy is flipping from outside to the inside. You call that a flippase. Now, if you look at other protein that is flipping from the inside to the outside, is also flipping, but you don't want to call that a flippase because it's a different protein, so you call that a floppase. Yeah. Some of the scientists have a good sense of humor, right? So this guy, the outside to inside is flippase, inside to outside is a floppase. But then you do have some integral proteins that can do both things. And you call that a scramblase. Scrambled eggs. Okay, so this guy can flip from either side, either this way or that way. So those are your flipping proteins. That's a cool naming of proteins. So here, looking more at your characteristics of your fatty acids, of your glycerol, phospholipids. So what do you have to identify here? You have to identify your glycerol, phosphate part, so phospholipids, right? Sometimes you have R group attached to the phosphate, sometimes you don't. But then you look here at your fatty acid side, you're going to have this chain R's there, so hydrocarbon there, and you're going to have two fatty acids connected to your glycerol, and then one of those carbons is going to be connected to your phosphate. This is what I said here. Okay? Considering the R groups, they can be occupied by several things. I'm going to find which ones are important for you to know. Okay? This guy here, look at this, is the major thing that you're going to find in lipid bilayers. So if it's a bilayer, you're going to find this guy. That includes liposomes, exosomes, microvesicles. What is the difference between an exosome and a microvesicle? Oh, it's basically the size of the vesicle. Microvesicles are bigger than exosomes. They're very, very tiny, like nanoparticle type of vesicle. Okay? Now, the enzymes that are going to hydrolyze the fatty acids, you call them phospholipases. Right? Just mentioned before briefly in another slide. Okay? So phospholipases are important for adding water and breaking down these bonds here, okay, carbon 1 and carbon 2, phospholipases. All right. So that's the story with your phospholipid. And now we're going to start talking about sphingolipids. All right. So what's going to happen here? You are going to start with your comparing to your glycerol molecule, but instead of using a glycerol molecule for a backbone, you're going to use a sphingosine molecule for your backbone. Okay, so let's compare here sphingosine to glycerol. Glycerol is quite simple. Okay, 3 carbons and OH for all the carbons. Sphingosine is a bit more complicated. Okay, you're going to have 3 carbon backbone, but here you're going to have this huge tail that is coming from palmitate. Okay, how many carbons? 12, 13, 14, 15 carbons over there. Big tail. And you're going to have a part of this backbone that is coming from serine. Okay, serine is an amino acid, right? So here you're going to have nitrogen part. So this is the first thing that is going to tell you that something is not the same as your phospholipid. Right? So this guy here has a nitrogen, so this huge molecule backbone is called sphingosine. And that gives rise to sphingolipids, right? Sphingolipids. they have nitrogen, okay? They all have this sphingosine backbone, and they are going to be important for some rigidity in the bilayer, and you're going to have to just differentiate what kind of attachment they have that will distinguish them for certain classes. So comparing here to your first fat that you looked at, which was a triacylglycerol, so the storage fat, glycerol phospholipid, you have glycerol backbone, right? 2 fatty acids, and here you're going to have your polar head. And now you get here to your sphingolipids, you're going to have your sphingosine backbone, you're going to have a polar head, and you're going to have some fatty acid that gets attached to that. Okay, so their composition is different. Now here, within its sphingolipids, you're going to have 2 subcategories. One of them is going to be sphingomyelins, another one's going to be glycolipids. Okay, so I'm missing an I here. Yeah, it is misspelled over there. Okay. So I know these facts get to be a bit complicated, but we're going to keep it simple as usual. Okay. So here, sphingolipids that you absolutely should know. You are normally going to find some sugar attached to those sphingolipids. So let's look at this guy over here. Okay. So here you have your sphingosine, here you have your fatty acid, and here for your polar head, you have 1 molecule of glucose, like right there. So this guy, to me, it looks like somebody with a head and 2 legs, okay? This head here, you could call it a brain. So brain, another name for brain is cerebrum. So you call this thing a cerebrosid, okay? So this guy is a sphingolipid that is a cerebrosid. This is what is written over here. Now, in comparison to another sphingolipid that has a lot of sugars, now you look at this thing and you say, this doesn't look like the little brain. It looks like a mess. So what do I call this thing? Call it a ganglioside. There's a gang of sugars over there. It's not just one sugar. Okay? So 1 little sugar over here, that is the glucose, that is your cerebroside. A lot of complex sugar is a lot of gangs of sugars that decided to get your head confused. You call that a ganglioside. Okay? And remember, this guy here has to have that nitrogen. Okay? So this guy is a sphingolipid. So 2 subcategories of the sphingolipids. Okay. Now, clinical correction. Why is this important? You need to metabolize those lipids in the body, and especially in the brain, okay? You have certain people that do not have enzymes to deal with metabolism of this complicated thing over here, okay, with this ganglioside. So a very important enzyme that you have in your brain that is going to help you to metabolize ganglioside, that is called gangliosidase, or here. Hexosaminidase A. This hexosaminidase, hexo has to do with 6 carbon sugars. So this is metabolizing, this thing is gonna need this enzyme. You abbreviate it as HexA, okay? If you do not have a working enzyme to metabolize this fat, you end up with a disease, with a syndrome that is called Tay-Sachs disease. Anybody have heard of Tay-Sachs? Yeah? So this is a recessive autosomal disease, is inherited, but you're only going to develop this if none of the genes coding for HexA works, okay? And you tend to see this genotype in Ashkenazi Jews, okay? So this is why if you are of Ashkenazi Jew descendants, it's always good to do a genetic test to see if you carry the gene or not. If you are heterozygous, you're fine. But if it happens that you have a child and the child is homozygous, this child is going to die. Because the accumulation of this lipid in the brain is going to make your whole brain to become fat. And this is neurodegenerative. And you're going to start losing body functions. Usually the child will die before age 3 or 5 at the most. It's a very sad situation. Okay? So lack of this enzyme, no metabolism of this lipid, and that is fatal. Now, these fats are also very important for formation of myelin sheaths. Okay, so remember here you have your cerebroside, your ganglioside, and you have also this modification called phosphocholine. Okay, so this is a very important lipid for myelination and neuronal neuronal nerve impulse. So another use for certain lipids is on formation of certain antibiotics. Okay, so some bacteria are very good at putting certain lipids together and making some forms of antibiotics, like polyketide is one example of these types of antibiotics. I know that I am putting out the time here. Okay, so here's just an example of these very complicated forms of lipids that are examples of polyketides. So those are antibiotic molecules. All right, so I'm going to stop here and we pick up on Friday.